we get experimental science. We're curious, non-judgmental.
Alright, I just wanted to show this off just as a little primer. These are praying mantises. I know, Lady Lara Croft, they don't look real, right? These are in fact real. But what I find really cool about these animals is that they shake. They move as if they were swaying in the wind. And apparently, at least the hypothesis is, that they do this kind of shaking and movement behavior as a means to make other insects truly believe that they're just a flower. Because the flowers also have a little bit of this like swaying movement as if the wind was, getting them, or just like general swaying of the plant. Now, I do not know what the underlying genetics of that Lady Lara Croft is. I've been looking online, and honestly, most praying mantis studies are about vision and trying to figure out how their very unique eye structure works, which is fair. It is a very, very cool eye structure. I don't know, we haven't truly figured out how it all works. All we know is that it's like a unique kind of 3D vision. There hasn't been anything done on this is an innate behavior. And so how they know to do this swaying, what gene is activated and what neuron is activated to be able to commence this swaying behavior, it's a really complicated question. It is not easily done. You'd have to develop a bunch of genetic tools in using this creature to identify that kind of question, but it'd be really amazing. Yeah, but Sam, what are the neurons that do that, right? What are the signaling molecules that control when it's time to sway and when it's not time to sway? What are those underlying features of it? I don't know. I would really like to know, what is that, too? The fundamentals underneath this. And, it is not easy. You'd have to develop tools that can mark neurons in the bodies of these creatures, CRISPR genetic engineering, maintenance of lines. It is really difficult. Even more difficult, Sam, is because how you can culture them is stupid hard. Because what you do is you have them hatch in a large container where you have a bunch of them. And Sam, it seems like they do not do well if they're isolated. They need to have a lot of different creatures with them. There's a social element of their behavior. And then, if you don't separate them out, once they hit a certain age, they eat each other, which is, I think, truly incredible. That they end up eating each other. So this is the regions where they're endemic to. So these are just general praying mantis locations here in yellow. So, there's praying mantises in the US, South America, Africa, a little in Europe, Asia, some in Australia. The ones for our particular orchid mantises says rainforests of Southeast Asia and Indonesia. Although you can get them in pet stores. Apparently there are some pet stores that have the proper permitting for these kinds of things, which is interesting. Yeah, you can't. There is still this very social element to that. They do much better if there's signaling between them once they've come out, and they do better once they've hatched together. But then, chat, it doesn't even have to do with the density in which the size of the container that they have. If you just made the container bigger and bigger, would they just not eat each other? No, it just seems like they become territorial and they just end up eating each other. How do I know this, Lady Lara Croft? Balint, this is a very strange thing for you to just know out of the blue. Surely, I'm not calling you Shirley, you don't have experience in this, right, Balint? No, in fact, Lady Lara Croft, I do have experience in this. We actually had a side project in the lab that was trying to grow up and start doing some kind of fundamental genetics in praying mantises. Mantid wasn't the one in charge of the side project. We were trying to culture them and getting stuff started with their cultures and trying to make sure that they'd be doing okay. But it actually ended up being where, someone else was in charge and he kept thinking it was funny that they just ate each other. And so there was a lot of that going on, unfortunately, in the background, which is not ideal. It's kind of, questionable of why things like that happen. But yeah, they will eat each other. But it was a cool idea to try to get some fundamental genetics together for the animal. The most I've seen, again, Lady Lara Croft, is the leading lab is at the University of Newcastle in England, and those are the ones that are putting 3D glasses onto the praying mantises, red and the blue, goggles, 3D goggles, to test their vision and figure out what kind of 3D vision these creatures have. It's really, really remarkable. They stop eating each other at some point in their development. So glued with gold, it's more that it's later on in development, is what it seemed like when we were using them. And that's what the literature suggests too. Early on, they're very social and they hang out and they're totally fine. And it's they hit after hatching a certain molt stage. And it seems to be variable between species of what molt that they start having this carnivory towards each other. And then they just go after one another and eat each other. Yeah. And I don't, it must be some kind of territorial density question, but in the lab we did try expanding that container, but I mean, I think what it is that room is pretty small. We're talking about 3D glasses on praying mantises because we rabbit holed and got excited, so that's what we do. And I think that's important to note. There is no regulation really on caring for insects in research labs, unlike mammals. But this is not a one-off thing. It's not that you glue this onto the creature and then it dies. This is actually a special kind of glue. Beeswax and a little bit of honey, and I think there's 1 or 2 other compounds in it. But what's really nice about it is you can take it off pretty easily and not hurt the animal. I don't know if you can see right now, but there is the red glass and there is the blue glass. So again, they're seeing in 3D, like with your classic 3D glasses. In order to test, like if you show them on a computer screen, those 2 colored balls start moving around. And then when they overlap, that should be 3D, like when we see in a movie theater. And then you can try to grab it. And actually, they do too. So, Andrea, this is the brain. And then that line running in the brain is the neuron. And it's 2 neurons, actually. So it's on either side. So it's symmetrical. So if you cut the animal's head in half, it's a mirror image on either side. So you have it going from the eye into the center of the brain, where it gets processed as what's called the fan-shaped body. That's the visual learning and memory center of an insect brain. And there's a bunch of more neurons here, Andrea. It's just they aren't glowing, they are not marked. The only ones that are glowing and marked is that one here that their researchers are looking at. And actually, in fruit flies, you have the technology to do marking of any neuron, right? So like 100,000 cells, you can mark any of them. The tools are not as developed in praying mantids, but it's still really remarkable that something like this is possible, I think. So, Andrea, every neuron in the brain of an animal has a particular gene that is activated in that neuron. If it's not, then it's not a special neuron. It might have a gene that is expressed in a couple of different neurons. Like this neuron, this neuron, this neuron all have this expressed gene, but usually it's something very specific per neuron. If you figure out what that is, you can hook onto that expression marker a green fluorescent protein, meaning that in every neuron of that type, you'll also express a glowing compound. GFP is called green fluorescent protein, and it is from jellyfish. And so you can turn on this thing from jellyfish in the brains of insects and mark a particular neuron. And so, Andrea, it's much more complicated than that, but that's essentially the crux of it. Alrighty, so you have a neuron. Here is this neuron in the brain. There's this branching pattern. There is the dendrite, the nucleus. Here is our neuron. This neuron in the brain of the animal is different than this neuron, which is different than this neuron. They're different, and it's not that they're just like physically different. It's that there are different types of neurons. They do different types of signaling. Does that make sense so far? That there are fundamentally different types of neurons that do different things in the brain. Like one thing might help detecting of odors. Another thing might help detecting of movement, visual cues. So the thing that determines what that neuron does is a gene. So in the DNA, if this is our strand of DNA, there is a region here called the promoter. With it for any gene, there is what is called the promoter, and then there's the gene itself. And so what ends up happening is that this gene is turned on. You then make mRNA, and then you make a protein, and then stuff happens in the brain from that. Whatever. Catch is that this promoter is specific to particular things in your body. Meaning, if you have genes that are supposed to be turned on in your liver, that means there are these things called promoters that tell your cells this is only supposed to be on in a liver, right? Because every cell in your body has all the DNA that every other cell has, right? But how do you know that a neuron is a neuron and a liver cell is a liver cell? It's in part based on this thing called the promoter. It makes it so that each cell knows what is supposed to be turned on and what is not supposed to be turned on. So again, it gives it it's what makes your liver not have heart cells in it, or lung cells, or brain cells in your liver. You only turn on liver cells. Like the tags on the spine of a library book. Yeah, a little bit like that, yeah. So each of these neurons also have promoters. So they have genes that are turned on in them that just make them a neuron. But how do you make a specific kind of neuron, right? So that instead of just having any kind of neuron, you want particular kinds of neurons. That is done by these extra promoters that are in each of these neurons. So each promoter in a neuron will not only tell the cells that, hey, you're a neuron, but it will tell them you're this particular kind of neuron. This is your job. I am assigning you a job, and that job is to do this thing. Your job is to detect visual cues. Your neuron job is to detect smell. Your job is to detect water. Like anything like that, Andrea, right? It's that promoter that makes you figure it out. And so then, Andrea, what you can do is once you figure that out, once you identify what the promoter is, you can then use it to do genetic testing. So what you do, and I think this is a really remarkable trick, and it's been done in flies, mantises, mice, human cells, you name it, everything under the sun. You take this promoter and copy it. So you put a second copy of it into the genome, but you do not hook on the same gene. So the gene here that tells this red cell to do visual stuff, you don't copy that over. You only copy over that particular part that tells it that this is where it's supposed to do stuff. You then hook on a new genetic sequence. So you've copied the promoter, and then you hook on something called GFP. Or it could be RFP or whatever. It's a glowing protein. This glowing protein will be turned on anywhere that this promoter turns on its gene. The catch with this is it makes it particular. So you've already identified the neuron of interest, right, through this promoter, because this promoter tells this neuron that this is what you're going to be doing. And so now you hook this promoter to a glowing protein, and everywhere that this is turned on, which is only in red neurons, you'll now also have green turned on. And so you can track the neuron visually. Essentially, you're identifying where does the GFP, so that you are actually injecting yourself. So you can do genetic engineering and insert this genetic element into the genome. It originally comes from jellyfish. So green fluorescent protein was discovered in jellyfish originally. There's also what is called RFP, that is red fluorescent protein. There's also CFP, that is cyan. There's actually a bunch of different colors, to be honest with you. There's tons of different colors. And so as long as you hook up this piece that tells you that, you know, this what this red neuron is supposed to do, you put that same genetic element to your GFP and it'll be turned on in the same spot. And so you can track that neuron in particular. So that's how you get this kind of image. So here, imagine this long neuron that you're seeing in the brain as that's the red neuron, right? And now that red neuron that you couldn't see, you hook that green fluorescent protein to the promoter and you make it visible. So that's how they do this particular experiment. And actually this has been done, like I said, in a bunch of other insect species, in cells, in mammals. The same principle applies, because in your cells, the principle applies, right? That's how your cells know that in this cell, we're going to turn on, you know, neuron cells, and in this one, we're going to do liver cells. There are these, like the beginning sequence of it, like we said. So like you've got the promoter element here. And glue with gold, this concept applies not just to neurons, but any kind of cell. Any cell it applies to. This glow here is only visible under a certain wavelength of light. You have to shine a wavelength of light that is 488 nanometers in wavelength at the tissue to make it glow. It is not glowing under regular wavelength of light. So, Shy Gamer, you'd have to hit all of your cells, like follicle cells in your hair, and then you have to stand under this particular wavelength of light. Now, the catch is, this wavelength of light can cause cataracts in your eyes. If you were to look at, like, straight, like, and this has actually happened before, I've had colleagues who've done this number, looked into the laser and destroyed their eyes, filled their eyes with cataracts, and had to have like emergency surgery from going blind. So usually you have like a see-through plexiglass thing that's covering it that's a different color. So like not quite clear, but it's like yellows, and that absorbs that wavelength of light. Pretty wild that they're using it for that kind of technological stuff, right? But yeah, that's the kind of end game right now for it. Or at least that's how they wrote their paper. I don't know if that's actually the end game of what they want to do, because it's not you know, you just have to say it for funding. I get the sense that they're just trying to discover stuff because they're actually interested in discovery versus trying to make Mantis robots, but